And this was proven to us by the statistically significant result as well as the marginally significant result. And I would definitely suggest pursuing a future experiment in terms of identifying if there is a threshold and then at what concentration that actually occurs. So overall, being part of the Tiny Earth Project has really allowed me to work on my scientific writing and critical thinking as a student. I'm a sophomore in my undergraduate career, so this was kind of my first dipping my toe into the research field, and I'm excited to continue that at the University of Wisconsin-Madison. And also, like I mentioned before, this, studying novel antibiotics and antibiotic production is such a novel field, and I think it's really cool and interesting to be a part of, that first steps in the research of this field. And lastly, at the University of Wisconsin, we have this phenomenon called the Wisconsin idea. That is that students apply what they learn in the classroom to what they do outside the classroom. It sounds sort of trivial, but we talk about it a lot. And I was able to see that through this process, through the Tiny Earth Project, I was able to do that and really kind of build on something that I probably wouldn't have studied so in depth in an introductory biology course. So I thank everyone for listening today. And if you have any questions, please let me know.
Thank you, Julia. Any questions?
You can also post your questions in the chat space and we can redirect them to Julia.
Yeah, I had a question. This is Danielle. I'm curious how you arrived at the concentrations that you did test of sodium chloride.
That's a good question. So actually this year, due to the pandemic, we weren't able to actually go into the lab and do all of these steps. So we actually just used the data from last year and previous years. So I don't entirely know why they decided on those concentrations. I wish that I was able to have done that and been a real part of it, but unfortunately we weren't able to get into the lab for the majority of the year until the end of the semester. And we did some PCR analysis, but that wasn't specifically what I was talking about today. So.
Anyone else? Well, thank you, Julie. I would also say grace under fire, hearing that loud noise, recording and all that kind of thing. So kudos to you. And
thank you.
And sorry about that.
No
worries.
It's a group thing. So just thank you very much for participating.
Thank you.
Our next speaker is Austin. Austin, are you here?
Yeah, I'm here.
Okay, great. And Austin, I think you have PowerPoint slides, right?
Yeah, I'm gonna skip around in that just for the sake of time, but yeah, I do have the PowerPoint.
Okay,
slides.
then you just tell me next slide or scroll, and I will. And I am going to. I don't know if you can see my little timer here, and you can see my screen and the slides.
Yeah, I can see the slides.
All right, excellent. The floor is yours.
Alrighty, so my name is Austin Minton. I'm an undergraduate researcher at Kentucky Wesleyan. I've been executing the Tiny Earth Protocol for approximately 2 years now, and I've been analyzing bacterial isolates found in the soil by a past class. So I'm going to jump to the 4th slide and just skim my methodology really quick. So I took 21 bacterial isolates. There was freezer stocks made of them, and I plated them on their predetermined growth medium utilizing the method of streaking for isolation. I used R2A, PDA, and BHI agar. And then I utilized 9 escape pathogen safe relatives for antibiotic resistance testing and plated them on LB agar utilizing that same method of streaking for isolation. And then I began my escape testing utilizing that method of patch plating. These 2 figures here just display that the top one here is my streak for isolation. This particular isolate is JSR2A5. So you heard the isolate, the type of agar in the isolate ID. And this bottom figure here just displays my escape testing with 4 isolates on here. So I'm going to go to the next slide. We did some further characterization with some biochemical testing, and we derived results for all the isolates that we deemed antibiotic producers, which was 13 of the 21. And we continued with some molecular analysis with PCR on the 16S rRNA gene and confirmed it with the agarose gel electrophoresis. And once these were confirmed, we sent them off for Sanger sequencing and put these in a blast search to determine the likely genus of each isolate. So just the next slide. This is just a big table of my escape testing. We used 9 safe relatives. And if you look at these, Bacillus subtilis, Enterococcus raffinosus, Staphylococcus epidermidis, and Mycobacterium smegmatis are the 4 gram positives of the 9. And we found that 8 of the 13 produce antibiotics effective against only these gram positives, while the 5 others, JSR2A5A, and the bottom 4 produce against gram positive and gram negatives. So showing this sort of broad spectrum antibiotic production. And the next slide. As I stated, we got biochemical testing results for all 13 producers. Looking specifically at JSR2A5 again, it was negative for both lactose fermentation and amylase synthesis. It was deemed gamma hemolytic, and positive for both catalase and oxidase synthesis. And then my next slide. This figure on the right just displays what is shown when we put a sequence into BLAST in the sequence coverage. This is also JSR2A5 again. And you can see here at the top of the right figure here that Genthenobacterium was the top genus match for this particular isolate. As a whole, on the left, you can see that 10 of the 13 derived Pseudomonas was the top genus match, while 3 others, Genthenobacterium, Arthropacter, and Flavobacterium, were the other 3. But as a whole, all of these genuses are found abundantly in the soil. And then just to conclude, I took 21 bacterial isolates from a past class, and they were deemed antibiotic producers by the past class. But when I tested this characteristic, only 13 of them actually displayed that. 8 of them produced antibiotics against only the gram-positive safe relatives of the escaped pathogens, while the 5 others showed this sort of production of the broad-spectrum antibiotics as it produced against gram-positive and gram-negative safe relatives. There were diverse biochemical testing results as a whole for all 13 antibiotic-producing bacterial isolates. 10 of the 13 derived their likely genus as Pseudomonas, while there was 3 others. As a whole, they're all abundantly found in the soil, so those results weren't necessarily surprising. And then looking back at JSR2A5, its ability to thrive in R2A agar, its unique morphology, as you saw, it appeared violet, its escape results as it was positive for antibiotic productions against both Staphylococcus epidermidis and Mycobacterium smegmatis. Its biochemical properties, as it was negative for both lactose fermentation and amylase synthesis. It was gamma hemolytic and positive for both catalase and oxidase synthesis. I believe the likely antibiotic it is producing is similar to a derivative of penicillin. And I plan to test this by first entering my results in the TinyEarth database, sending isolates off to the TinyEarth Chem Hub, and then starting with some chemical extraction of the purified product, which I began, but with COVID closing the school down, I kind of had to stop that. And then with this purified product, I plan to look into some bacterial inhibition testing, and then also look into some host toxicity testing with eukaryotic cells, and then some structural characterization with chemical analysis. The next slide is just my acknowledgements of everybody that basically contributed to this research as a whole over the past couple of years. And then I open the floor to questions. Thank you guys for listening.
Thank you. Any questions?
Austin, I was curious if you had a hypothesis as to why you saw a reduced set of antibiotic producers.
So I looked into that. I mean, I've thought of multiple things. It could be just this was done by like an introductory course. As I'm a junior now, so I'm a little bit more advanced in the protocol itself. I think it could be due to just bad, I guess, recording of the results, or just basically just not executing the protocol correctly, or just something is simply maybe something going wrong when preparing the freezer stock, because they were in the freezer for about a year. So yeah, there's like basically an infinite amount of ways that I could think about what caused that. But yeah, that's what I've kind of concluded so far.
Yeah, I think we find that a lot in science, but especially in biology, it can be super tricky to repeat. And so making sure that you're using the right controls and understand your variables, but awesome. Thank you.
Appreciate the question.
That's a good point, Austin. We noticed that as well when we freeze our samples and then pull them out a few months later. Some of them do end up losing their antibiotic activity. All right, thank you. Yeah, and I see more people writing in the comments that that is a known phenomenon that we're noticing, several of us. All right, thank you. We'll go to the next presentation, and that's by Bryce. Bryce, are you here? Is Bryce not here?
I'm looking too to see.
Okay.
I don't see him already. Yeah.
I don't see him. I'm going down the list. Maybe we come back to him if he joins in. Amanda?
I'm here.
Okay, great. Amanda, let me just pull up your presentation. Sorry, I'm doing this the old school way. For some reason, the other one isn't pulling up everything. Here we go. Amanda, got yours. And I'm just going to share. Okay, and just tell me if you need me to scroll in one direction or the other. You can see it now?
Yes, thank you.
Excellent. The floor is yours.
Thank you. So hello, everyone. I'm Amanda Brechko, and I'm a senior undergraduate student at Campbell University in North Carolina. Today, I'm going to be presenting on the identification of potential antibiotic-producing bacteria from soil at Campbell University. So while antibiotic resistance is affecting all areas of healthcare, an increase in the number of antibiotic-resistant organisms present in the neonatal intensive care unit, especially vancomycin-resistant enterococci, presents an increasing risk to neonates. And since I'm really passionate about helping our youngest population, I wanted to contribute to defeating this crisis. So the goal of this study is to isolate potential candidates of new antibiotics to combat this growing antibiotic resistance problem, especially among newborns. So if you don't mind moving on to the methods, topsoil was collected from 2 different sites. Site 1 is the edge of a woody area at the bottom of a hill, while site 2 is above a runoff into a creek. Do you mind zooming into the methods for me at the bottom left? Thank you. So a series of 4 10-fold serodilutions were performed on each of the 2 soil samples, and soil isolates were cultured on plates of potato dextrose agar, which is PDA, and then tryptic soy agar, TSA. The turtle tracks method was used to test the susceptibility of Enterococcus raffinosus, the safe relative of the escape pathogen Enterococcus faecium, against potential soil isolate antibiotic producers. And then pure colonies were obtained using the T-streak method for the 16 soil isolates that showed potential antibiotic production. And then polymerase chain reaction, PCR, was performed to amplify the 16S rRNA gene of the bacterial DNA, and agarose gel electrophoresis was performed to determine the success of the PCR product. The DNA of successful purified PCRs were then sequenced by Eurofin's MWG Sanger sequencing, and the sequencing results were analyzed with the 4-peak software program, and high-quality sequences were run through the BLAST database to determine the soil isolate's closest relative. So moving on to the results in the upper middle column, Figure 1 displays the spread plates of the soil dilutions. So as you can see, colony forming units per gram of soil was found for the 10 to the negative 3 dilution plated on PDA, as well as the 10 to the negative 2 dilution plated on TSA of the soil sample from site 1, and the 10 to the negative 2 and 10 to the negative 3 dilution plated on PDA, as well as the 10 to the negative 2 dilution plated on TSA of the soil sample from site 2. And then unique bacterial colonies from these spread plates were picked to test for antimicrobial activity as seen in figure 2, which displays the results of the turtle tracks method. So if you don't mind zooming in more on figure 2, you can see that the 16 soil isolates underlined in red inhibited the growth of Enterococcus raffinosus at some level. And then the success of the PCR product of these 16 soil isolates is shown in the next 2 figures down below. All but 2 soil isolates, ALB11 in well 4 and ALB13 in well 5 of the gel image shown on the left had a successful PCR product, indicated by the bands around 1,500 base pairs. And then moving over to table 1 in the upper right corner, the putative identification was found for 12 soil isolates, and colony morphology was used to characterize them. There are disparities in characterization among different soil isolates with the same putative identification. So as you can see, soil isolates ALB 5, 18, and 19 were all putatively identified as Bacillus wiedemanni, but the colony morphology of ALB5 was different from that of ALB18 and ALB19. And then another disparity is soil isolates ALB39, ALB50, and ALB51 were all putatively identified as Burkholderia contaminans, but the form and color of the soil isolates were different from the actual form and color of this bacterium. So the conclusions demonstrate that there was no noticeable difference between soil isolates cultured from soil samples collected at the 2 different sites. However, media did affect soil isolate growth, with soil isolates growing better on TSA compared to PDA. So these identifications are all still preliminary, and further screening of the 12 soil isolates will be performed in the next phase of this study. Thank you.
Thank you. Any questions for her?
I'm on a roll, so I'll keep going. My question was related to the different morphologies despite where they were identified, and if you had any hypotheses about why they looked different even though they were coming back with the same result.
So I'm not really sure why they're different. Maybe the length of the sequence that we put into the BLAST program, maybe since if it was a small length, the percent identity was a high percent, almost 100, but maybe if we had a longer length, it would have shown a different identification.
Okay, great. Thank you.
Our next presenter is Kylie. Is Kylie here? Kylie McCain.
I don't think so, Artie.
Okay.
Yeah, she was unable to make it.
Oh, okay. And then I will go down the list. Casey, is Casey here? Casey? No, Casey. Vincenzo.
Hello,
Oh, there you are.
I am here.
Okay, excellent. So hold on, let me just pull up your slides and then I will share them and you can tell me to go I will try to zoom in as you wish to whichever part of your poster. And I'm gonna set this, I don't know where this is appearing for you guys, but the floor is yours, Vincenzo, and you're coming to us all the way from Italy.
Great.
Hello, I am Vincenzo Somma, undergraduate student of biology from the University of Naples Federico II in Italy. The title of my presentation is Exploring the Antimicrobial Capacity of the Soil Microbiota in the Mountains of Gragnano, Naples. The abuse of antibiotics has led to the emergent problem of antibiotic resistance around the world. Italy is one of the most affected countries in Europe, and in particular, the region Campania is one where the resistance is widely distributed among the population. Microorganisms living in a healthy environment that can be an excellent candidate to find antimicrobial producers. So in this study, I identified for sampling a contaminated green area rarely frequented by people. The mountains of Gran Castello in the country of Gragnano, and this habitat makes my project unique. The sample was collected under hazelnut tree. The area was humidly covered by vegetal residues as fallen leaves and the presence of wild animals in the laboratory. The soil was processed following the methods of dilution and inoculate plating in two different media: TSA 10% and M2 cyclohexamide, that is a variation of H2A medium. After incubation, a total of 48 colonies were selected for the master plate, and successively the antibiotic screening was performed against two safe microorganisms: the gram-positive Staphylococcus epidermidis and the gram-negative Pseudomonas putida. It was observed that 3 isolates inhibit the growth of Staphylococcus epidermidis, and a second screening was performed in the research laboratory, but on this time against attenuated strain of human pathogens. As a result, as you can see in figure 1, only one isolate, the strain CN5, was able to inhibit Staphylococcus aureus. According to 60 in sRNA gene sequencing, the strain CN5 was identified as Bacillus zancobensis, 99.6%. The genus Bacillus is very known in the literature to produce metabolites with antimicrobial properties. The complete genome available on of the closest strain, Bacillus zancobensis, was genome managed using the tool Anti-Smash in order to identify the biosynthetic gene clusters for secondary metabolites production. This method used for very interesting to me because it helped me to understand how the analysis of the genome gives information about the synthesis of molecules probably related to the halo of inhibition observed in the screening. After the analysis, as you can observe in figure 2, the genome of Bacillus zancobensis revealed the presence of 10 encoding regions for the production of secondary metabolites, of which 6 are not yet identified, indicating that similar isolates belonging to these species can produce molecules with antimicrobial capacity. This strain CN5 is the ideal candidate for chemical analysis, and the future direction of the project in order is to identify if the strain is producing an already known compound, or if the antimicrobial action is due to the production of new molecules as the genome reverse. Regarding COVID, Tiny Earth was organized as a summer school, and we had the opportunity to complete all 4 laboratory seasons face-to-face, programmed without interruption. Thanks for your attention.
Thank you. We'll take any questions that people may have. That was the last speaker, so we can open up the floor for some questions for any of the other participants as well.
One of the things, maybe as a part of this, that I think certainly Artie and I have seen yesterday and certainly today as well, is how amazing it is, the work that you've done under these kinds of circumstances with this pandemic. Microbes, as we know, they don't always lend themselves to these reconfigured time points and time frames that we've had to implement in our labs. So it really is just it's a remarkable thing to bear witness to. So, certainly want to make sure that we share that with all of you.
I have one quick question for Vincenzo. The organism that you identified it as the bacillus, wasn't it? I was trying to scroll through the slides as well. Do you know, is it something that's common to that part of Italy or Naples, or is its origin known to be from somewhere else?
Sorry, I didn't understand. Can you repeat your question?
No problem. I was just wondering, the organism that was identified as, is it something that's commonly seen in the soil from that part of Italy, or is it just something that?
No, we cannot homogenize the results because the area is very different. Some of the students take the sample, for example, from the sea area, another from the river, another from the mountains. They were free to choose the best place for sampling. And that is I wanted to do like this, not otherwise we will have the same or common results. And also I wanted to see the difference among the different soils, to understand the cultivability of these microorganisms, to understand the aerobic microbiota present in the region, because they don't live just in Naples, they live around the surroundings of the region. So it's important because it's the sea area, it's a volcanic area, it's a different height. This was
I remember there was another poster, but that was more from the water, if I remember yesterday's poster. They sampled more closer to the water.
Yes, the water, another from the soil, polluted soils. So depends of the results. In the case of we are presenting only the soils that were able to isolate some microorganisms with activity in the laboratory against safe relatives, and then in the laboratory where I am here now, we repeat these screenings, but against human pathogens attenuated, and then only to confirm if they are able to do something else.
Excellent.
Yeah, it's wonderful to see samples coming from these different sites. It really, really is. So
yes,
best of luck going forward with all of it.
Brian, I'm going to let you speak some more, take questions while I run over to the next session and set up over there.
Okay.
Thank you.
Brian, I wanted to add something that you said that I don't know everyone here understands about themselves, but just hearing when you guys speak about your science, it's so great that you don't just get results and accept the results. You've evaluated your results. You're looking to see that it doesn't quite make sense into what you observe when you culture them. I think that's so important for us as scientists to never let go of, that even when you're far into your careers, that you're still looking at what you get and assessing it in the context of what you already know. And so I commend all of you for that. Thank you.
Yeah, absolutely. Yeah, no question about it. And just some of the fun things, right? So you especially during the time of COVID-19, you get results through blast analysis, and it gives you an idea of what the genus is and then what the gram reaction should be, right? But then you're working with a culture that's a week old because of safety reasons and how your lab is set up. You can't go to the lab every 48 hours and different kinds of things. And it's just been an interesting experience in so many different ways to try to do all this during a pandemic like this one. Yeah. So anything else? Okay, well, we have this great keynote that's starting in 17 minutes. Really looking forward to that. And thank you all so much for your contributions, because it does all matter. So we'll see you in just a few minutes. Thank you. Bye, everybody.